Hello and welcome to this special EULAR review podcast brought to you by the Lupus Forum. My name is Ed Vital from the University of Leeds, and today I'm joined by Professor Chris Edwards, who's a consultant rheumatologist at University Hospital Southampton and Honorary Chair of Clinical Rheumatology in the University of Southampton. Welcome, Chris. How was EULAR for you?
Really good, and I'm looking forward to the discussions. Lots of things to talk about.
Yeah, exactly. There was an awful lot, and we've got quite a lot to work through. So we're going to go fairly quickly through these. So we're going to talk about what we found most interesting at EULAR. There's lots of lupus content, and the most important is all listed in the highlights brochure if you want to find other abstracts. There's over 70 in there from across the conference, covering a wide range of different topics and different therapeutics. And it's also worth knowing, if you want to look at abstracts in a similar way for rheumatoid, psoriatic arthritis, axial SPA, then there's a similar highlights brochure and podcasts for those disease areas on the Immune-Mediated Inflammatory Disease Forum. But I think it's time to get right into it. So we'll go to the first couple of abstracts that we've selected here. And this was some of the talk of the conference, really, which is CAR-T therapies. So there's some update to this. So if you haven't been following this, the basic principle is that we've known for a long time that although rituximab could be quite effective for lupus, it didn't deplete B cells deeply enough, and that's one of the reasons for the variability in response. And many of the attempts at new therapies since then have been trying to deplete deeper. There's a similar problem in hematological malignancy, so some of the innovations come from there, including this one. And the basic idea of CAR-T therapy is, instead of targeting CD20 with a monoclonal antibody, you target CD19. CD19 goes a little bit further into plasma cells, so it's a bit broader in what it targets. And instead of just being a monoclonal antibody, what it is one of your own T cells reprogrammed with a receptor that targets CD19. So then those T cells are given back to you, and they will go around depleting B cells. And that is a more efficient mechanism, especially when you have to go into other tissues. So that's the idea. Deplete deeper. The initial case series on this were reporting that very high rates of remission, and also that people were stopping all other therapies. So that was creating a great deal of excitement as to whether this quality of response was so much better and quite durable, and maybe even in some people kind of curing their autoimmunity. So what have we got updated at this conference? Previously, we've had quite small case series. Here, we've got a couple of other things. This is another study called Castle from the original Erlangen group, who published the first case series, but just more patients. So far here, we've got 5 patients with SLE, 1 with scleroderma, and 1 with inflammatory myositis. Data's pretty early, but there's one important thing you get from early data, which is safety. Because the thing about CAR-T is it can cause a couple of fairly significant toxicities. One is a cytokine release syndrome, which can sometimes require treatment in intensive care and need treatment with tocilizumab. The other is a neurological syndrome called ICANS, which can be quite variable, but can become quite serious. Those are big in hematological malignancy, and the bottom line here is those weren't really problems in these rheumatic diseases. It wasn't in the previous case series, and it wasn't here, none of the serious stuff. Early response data here suggested people were all getting some level of response, but it is quite early, some of them only 6 weeks, so we wait for longer for the efficacy there. And then I'll just go on. And just on the screen there, you can see those main results there. So there's the details on the patients and their safety in the table. On the figures, you can see the CAR-T cells as they're infused. You get a peak and then they disappear. And the B cells get depleted and start coming back about 3 to 4 months, which is quite early B cell return. And you can see some change in leukocytes there as well. You can get a little bit of neutropenia like you do with rituximab after this. And then I'll just mention another CAR-T related abstract, which is so one of the other feelings about why targeting B cells might not work is that some of these antibodies are made by longer-lived plasma cells, which won't die if you just target B cells. Some plasma cells have got CD19 on them, but not all. BCMA is a receptor that's expressed quite widely on plasma cells. So if you make your CAR T product target BCMA, you're depleting plasma cells. And if you do what they did here, which is target BCMA with some CAR T and also target CD19, then you're hitting both. So that's very, very broad depletion of B cells and plasma cells there. This was a study done in China with 10 patients with lupus nephritis. Again, they say these severe fusion reactions of cytokine release and ICANS didn't occur. At 9 months, protein creatinine ratio was down by about 2/3, and all the autoantibodies essentially disappeared. So again, we're still on small numbers of patients, but it seems interesting. It seems promising, but there is a lot of talk about what this really means, isn't there? What did you think, Chris?
Yeah, I suppose I came away because the more I see it and the more I see trials showing, I mean, albeit small numbers at the moment, the more I see trials that don't show scary side effects, the more reassured I am.
Yeah.
You know, a number of us have been having conversations about taking part in some of these trials, and you're trying to weigh up the risk for your patient group and thinking, should I be taking part? Is this a reasonable thing to do? But this is relatively reassuring, and some of the other data we see is reassuring about that. So I thought that. I also felt there's a lot of fuss in general about CAR T at the moment, and there was at the meeting. So lots of people are discussing it. Lots of people are talking about it. A lot of companies are getting involved in the space as well. But I guess the uncertain things are how long do we go? Do we really know that people will stay without treatment, that their disease won't come back? Will they all come back in a couple of years? Will they all come back at 4 years? Will some not? Those are the uncertain bits that will make a massive difference to whether this is something we do a lot of, because this is a big deal to do and it's an expensive thing to do. So it's got to be safe and it's got to keep people well and without treatment for prolonged periods of time to justify it, I think.
Yeah, so exactly. So I've always said that we need to deplete deeper. So the fact that deeper depletion gives you a better clinical response, completely on board with that. There are some other drugs that will give you deeper depletion. I think one of them we're going to talk about a bit later, but anything that gives you deeper depletion should be good. The bit I take more issue with is the idea that this could be a cure for lupus, because that basically, theoretically, the concept is that B cells kind of went wrong by accident. And that's how the lupus started, because of the cells going wrong by accident. And if you correct that mistake, it won't happen again. And to me, that's against everything I know about lupus. Everything I know theoretically about lupus is kind of that B cells didn't go wrong by accident. They went wrong because of like innate abnormalities, like interferon, or even things that aren't even in the immune system, like apoptosis and cell death, which is in every cell in the body. And those things aren't going to disappear because you depleted B cells, those problems. So my guess is that would predict that you're going to, eventually the B cells will become autoreactive again and you get lupus again. Saying that, that's no means guaranteed, because we all know that people can walk around with many of these abnormalities, ANAs, interferon activity, lots of other stuff, and be completely healthy, what we call risk population. So, what's it going to be? We don't know. But I think there has been some talk about a few cases of relapse being seen after this therapy.
Yeah, well, there was some discussed at one of the sessions, weren't there? I saw Dulcek mentioned, I think, a myositis patient that had relapsed, and they'd retreated them with the remainder from the batch of CAR T that they had. And then there was some discussion about, if you don't respond to the CAR T the second time, maybe there is some immune response against the CAR T cells that you're putting back in. So maybe it won't work as well a second time, and you have to have a different strategy for if people relapse. But there's probably no way to know this at the moment, is it? You just have to run with the trials, see more patients, understand safety and how many come back again.
And the other thing I think to be aware of is that this is only there are many technologies being developed. So CAR T, there are lots of things that are CAR T, different targets, different ways they work. There's also what's also generating a bit of chat, and I bet we'll hear a lot more about it next year, is bispecific T-cell engagers. In other words, giving the programmed T-cells, you give a monoclonal antibody that'll link a B cell and your own T-cell together. So the injection is just a monoclonal. It creates the reaction in vivo. And of course, there's just better CD20s.
Yeah, but even amongst what you've shown, these two studies, so we have let's remove the cells that have CD19. Then we have technology saying let's remove those cells plus some others as well. And you can imagine more and more bespoke approaches where you take out different parts of the important bits of the immune system. And I guess that will grow over time as well.
And it all needs to be proportionate. I mean, at the moment, this is being thought of as a therapy for the most severe patients, but exactly where its role is in the clinic will depend a bit on all these things we've discussed. Overdoing plasma cell targeting can come with some definite side effects in terms of...
and then that's going to have a really big effect depending on your cohort. You can imagine there are parts of the world or certain populations where disease is really, really terrible, and this sort of thing may be justified. And then there is slightly more benign cohorts that might not need this therapy very often, or if it turns out to be more widely used. But it is also, fair to say, it's also interesting to see it in multiple other areas. Although we're talking about lupus now, the fact that you can give this to really untreatable scleroderma, and that might have a benefit. So, some of the other indications make it attractive as well, not just, just the lupus story.
Okay, let's go and talk about some of those other options then. So next one, you took a look at this one, didn't you?
So the deucravacitinib story. I mean, we've been looking at JAK inhibitors for a long time in a number of disease areas. Here's TYK2 inhibitors, so JAK1, JAK2, JAK3, and then TYK2 is the fourth member of this class of signaling molecules. And it's an evolving story of we're getting bits of the trials bit by bit by bit. This, of course, is lupus. So this is from the phase 2 Paisley trials, and this is Eric Moran presented this. But this is being trialed in other disease areas as well, of course, at the moment, like Sjögren's syndrome. And it showed a number of different results that you can see mentioned here. I don't think there's anything particularly new to say. It's just more to say a continuation of the story. Another target in a class that we're sort of slightly familiar with, but bringing TYK2 inhibition to lupus.
This one was kind of looking for patient subgroups, and they looked at things like age, steroid use, SLEDAI score, disease duration, and didn't really see any difference in any group that benefited the most. I noticed that there are still some things I'd like to know more about, like what about which organ was responding or different antibody subtypes. But I think we need the phase 3 for that because the numbers are a bit small. And then another one, which is a kind of biomarker development on an evolving story, is this one that I presented, which is about the Nobility trial. This is obinutuzumab, which is one of those next generation CD20s. So basically, rituximab's class 1 CD20, obinutuzumab is class 2, which is what we generally think is better and more modern. And class 2 CD20s, they bind to the CD20 at a different kind of orientation. And they've got a glyco-engineered Fc domain that's better at engaging the secondary killing cells. And they can also kill the B cells by themselves. So basically, class 2 are more potent at killing B cells. And so this trial's already been presented that showed that obinutuzumab plus standard of care in lupus nephritis was better than standard of care alone, standard of care being mycophenolate and steroids. So that's already been shown. It demonstrates its efficacy. This particular abstract was about the biomarker part of it, basically using a method that I used in my PhD, which was a highly sensitive flow cytometry method so that you can follow different B cell subsets through treatment. So the main conclusion is the OB was given at 0 and 6 months, but then the clinical response was at 12 months, 6 months after the last drug exposure. And the total follow-up was 24 months, so 18 months after the last drug exposure. And the main things you could see, depletion appears to be better than rituximab. It's not side by side, but to my eyes, it's better depletion. The clinical benefit continued to be different between the treatment and placebo, all the way up to 24 months, perhaps even widening, even though the treatment had finished at 6 months. So, long-term clinical benefit. The repopulation pattern is what you see on the screen there, which is, so what we learned from rituximab is that after you give it, the ideal pattern for a certain patient is that the naive B cells come back, but the memory B cells and the plasmablasts remain lower than they were at baseline. That's a kind of good pattern that forecasts a good response for an individual patient. And here you can see that pattern at a
That's
group.
that pattern, isn't it? That's exactly what you can see.
Yeah, so at a group level, you can see most people got that desirable pattern. And there was a little bit of evidence here that those with the best depletion got the best responses. But again, that got into a bit small patient numbers.
Yeah.
Okay. What have we got next? So here, this is a review on rituximab in real world use. You looked at these, didn't you?
Yeah, I did that. Well, there's a couple about BILAG biologics registry, which I'll come to in a moment, but it's also nice to look from other parts of the world. So this is data from colleagues in Japan. They've got a registry called the Lupus registry, looking at lupus nephritis patients, and they were looking at rituximab against standard of care. And they followed these patients out for a period of time, looked at retention rates, staying on the drug, how much they did that, what happened to the SLEDAI scores over time, and adverse events as well. And I guess, I mean, I felt a little bit disappointed about it because it kind of says that the rituximab was no better than the standard of care. I suppose the optimist might say that it is saying the rituximab is at least as good as the standard of care.
Yeah.
But it didn't show, it didn't show outstanding efficacy. It didn't show anything bad happening either. But particularly there are a few infusion reactions, as you might expect with rituximab, but nothing more than that. I don't know. Am I wrong to feel slightly negative about it?
Yeah, I think I agree. So the interesting thing, I mean, there's a couple of things I thought. One was, as far as I could see, it was the rituximab plus glucocorticoids was one group.
Yeah.
The other standard of care, meaning mycophenolate or cyclo plus glucocorticoids. And those were about the same. And like you say, yeah, is that good? Is that bad? It's an alternative, but it's not an advance. But then I kind of thought, well, actually, I would do them together. I'd do rituximab and mycophenolate.
So maybe it was some
study that I just presented was obinutuzumab and mycophenolate, which I don't think is what they've done here.
Yeah, so maybe it's unfairly disadvantaging the patients on rituximab by not having the mycophenolate at the same time.
Yeah. Then again, of course, when we first started using rituximab 20 years ago, what people were saying was very much like what people say about CAR-T today. Depleted all the B cells, long-term response, don't need other treatments. And now, with the maturity of time, we realize, it can be a useful treatment option, but it's got its place amongst others.
Yeah. But it's also good, and we've got a few of these on some of the other. It's good to see data from other parts of the world and parts of the world that have different sorts of lupus sometimes as well. So
and real world is a lot messier than trials.
Yeah, always. Yeah. Okay. I think we're
next one.
Probably on to the next one. Okay, so there's a couple of these. So the background, of course, is the BILAG biologics registry has been running for several years, and it was set up mainly as a safety registry. And it was to put belimumab and rituximab onto it and follow it over time. Of course, there are other biologics and other treatments, and there is a smaller control group as well. And this was a nice presentation about the problems of neuropsychiatric lupus, all those different multiple manifestations that may occur within lupus patients. And this session resulted in a bit of discussion and some questions at the end of the presentation as well. The difficulty, of course, is this wasn't controlled, the data they presented. What they showed is here are a group of patients who have neuropsychiatric manifestations, and then we'll look at what they're receiving. We'll look at them receiving rituximab, and then we'll see how much less disease they have over time. And so it's very hard really to say, but the suggestion of the author was that this suggests that being on rituximab when you have one of these diseases is associated with improvements over a period of time and getting less neuropsychiatric manifestations and reducing steroids and some of those other things as well. But the questions were around, but you don't have a control group, so this is slightly difficult to be certain about. And that was the weakness of it, although it was a really nice presentation. And it raises what is an important part of disease and sometimes very difficult to treat, particularly the more serious manifestations.
Yeah, absolutely. And I mean, I suppose the thinking, optimistically about what it means about rituximab. One is that if you're outside the UK, one thing to know is the rule is basically you've tried other medications before. Before you get rituximab. It's not always adhered to because they can just be contraindicated, but you're meant to have tried either mycophenolate or cyclophosphamide. So then you might say, if you've had those, you're still active and you respond to rituximab, it's promising. But again, real world practice is messy. The other thing, though, I thought was that, and I think this was mentioned writing the EULAR guidelines, is that the evidence for anything else is this level. So belimumab, anifrolumab, and all the other trials that are still in progress generally exclude neuropsychiatric lupus because it's hard to assess and consent might be a problem, etc. So it's usually excluded. So everything's case series. And in the EULAR guidelines, that's kind of alluded to that most manifestations is belimumab, anifrolumab, licensed proven biologics. But when it comes to neuropsychiatric, there's a different recommendation that says cyclophosphamide or rituximab, which is case series evidence. So it highlights the gap in the evidence, doesn't it?
Yeah, absolutely right. But I guess it depends which manifestation we're looking at. Some of the really serious ones, if they're improving in this group, they probably wouldn't have improved without treatment.
Yeah.
Some of the milder things, lupus headache, well, that's going to come and go over time.
It's a very mixed bag, neuropsych.
And it's
exactly,
and it's going to be very difficult to attribute that improvement to rituximab in this sort of study.
So difficult study to do, but the best data we've got so far.
Yeah, I think so, and it's encouraging. Yeah, it's encouraging. Right, okay. Next one. Okay, so this is looking at discordance in clinical responses. It's essentially thinking about the difference between how patients feel with their disease versus the clinical scores that we can look at, and trying to see if you can find out particular populations, particularly groups of individuals that you might define by their demographics, by their disease activity scores, by their damage index, and associate. So this is less, although it's the biologics, the BILAG biologics registry again, this is less about rituximab, although many were on rituximab.
So they said that the clinical people who clinically responded by the physician's judgment, i.e. BILAG SLEDAI, 56% of them improved on their SF36 mental, and 70% improved on their SF36 physical. So there's some discordance there, isn't it? And what was highlighted was that the discordance on the mental component was associated with people who had lower SLEDAI score, East Asian ancestry, more damage, more socioeconomic.
Deprivation.
Yeah. And on the physical, you were more likely to be discordant if you had lower steroid dose and if your therapy was belimumab. Isn't it? So I guess it's just highlights this comes across a lot of trials, which is that drugs that seem to work on disease activity measures is often harder to show a benefit on patient-reported outcomes.
Yeah, and I think that's true in all diseases, isn't it? I mean, we see it in all of them. That's why people define difficult to treat disease. We measure, and in fact, maybe we'll come back to this. When you look at some of the new criteria for dividing up disease in different ways, maybe that will remove some of this difficulty. We're including this great big group of people, aren't we? We're saying everybody's symptom is the same. It's being caused by lupus, but some of it is driven by certain cytokines, certain cells. Some of it is being driven by different things completely and will not respond to us using particular therapies. Anyway, we might come back to that.
The thing that determines your quality of life. Maybe at the start of the disease, it's all about disease activity. But once you've had it for a long time, you've got a lot of other issues. You've got your steroid use.
And the damage.
You've got the damage. And also sometimes I think, when you talk to patient groups, one thing people sort of kind of say is like, I'm just having a different life from what I kind of thought I was going to have when I was 18. I haven't got the career I thought I was going to have. I haven't got the family I was going to have. I've lived with a disability, stuff like that. And drugs can't give you the last 10 years back. So Yeah, it gets more and more complicated what you need to do to improve quality of life. So yeah, as you said, the next ones we talk about.
Yeah, and I know to interrupt, but I really like these two. I'll tell you what I mean. I really enjoyed thinking about these, but forgive me, I interrupted you.
Yeah, no, that's right. So one of them is mine. It's my conflict of interest on these. But the first one is one from Japan. So what they did here was they tried to subset lupus patients, and they decided to do it on a purely immunological basis. Basically, do flow cytometry. We all know that lots of cell subsets can be up or down. You can have more or less Tregs, different subsets of B cells, plasmablasts. All of these things can vary from patient to patient. And so what they did was they took a group of lupus patients, 123 of them, plus 75 controls. They did flow cytometry on them all, and then gave that flow data to a machine learning algorithm that basically tries to cluster it into groups. You decide how many clusters looks about right, which they thought was 4, looks about right. And then once they'd got those 4 clusters, then they looked at the clinical data to see if these were clinically different. And cluster 1, they said this is a kind of similar. The immunology looks similar to the healthy controls, and the disease activity was mild. Okay. Cluster 2 was patients who were quite young, had a lot of double negative B cells. Cluster 3 was patients who had super high levels of plasmablasts, which is generally considered a bad thing in lupus. And indeed, when you looked at the clinical data, they had more disease activity in low complement. And cluster 4 they were marked out by higher levels of Tregs, which might be a good thing. And when they looked at the long-term follow-up of these people, they looked for how many relapses they got. And cluster 1, the mild one, no relapses. Cluster 3, the plasmablast severe one, 20% had relapses, and the other 2 clusters, 10% had relapses. So some evidence there that if you just sort patients out by their immunology, then you'll get different clinical phenotypes.
And you think actually, some of that if I think about being a registrar at St Thomas's, and you're seeing the patients years ago, and you're trying to work out what prognosis they'll have and how they'll do, and you're saying, okay, the people that do worse seem to have anti-RMP and they have anti-SM. They're not always the ones with the highest anti-double-stranded DNA, and they're more likely to get lupus nephritis, and then this group is more likely to do better. It's trying to convert some of that clinical nous that people tried to use into a science of defining groups. And therefore, it allows you to think, if I'm doing a trial, maybe I only want to be doing the trial in cluster 3, where they've got loads of plasmablasts, and they will respond to me getting rid of their plasmablasts. That's what's so important about it.
That's exactly, that's the really important part, I think, whether those are the people who need the right therapy. And that was kind of the motivation for the other one, which is one from my group. This is the PhD of Jack Arnold, and it represents quite a lot of work on his behalf. And well, this might be a strange thing to be saying on the Lupus Forum, but the idea is shall we stop talking about lupus? So the idea here was that actually lupus is quite a complex and heterogeneous disease, but it actually exists within a family of other diseases, Sjögren's disease, inflammatory myositis, scleroderma, these undifferentiated patients, mixed overlaps, all of this stuff in this kind of soup of related diseases. And sometimes the boundaries between them seem quite clear, but sometimes they're actually maybe not so clear and maybe not that helpful. Like one example we have was if you've got lupus and your main problem is arthritis, then you could have belimumab. But if you've got Sjögren's syndrome and you've also got arthritis, theoretically, you can't have belimumab, even though your problem is the same, and probably the immunology driving it is pretty similar too. So all of that suggests maybe the way we classify this stuff is not correct. So as the abstract we just heard just tried to classify it purely by flow cytometry. There've been a couple of other attempts before that use just immunology with the idea that let's just be immunologically pure and not worry about what the clinical features are. So the idea here was firstly, rather than just lupus, we're going to take multiple diseases and mix them all up and see how they come out. Do they segregate back into those diseases or something else? But also, this is going to be a hybrid of kind of clinical and molecular. So the approach was, first of all, there's two data sets here. There's one called Precise SADS, which is European, which is it's got over 2,000 patients in it, but about 1,300 that are relevant to this. And the other one was one of our own, a study called Definition that had 300 patients in, hundreds of variables. So the first part of this was to try and think which of these variables are the priority, like which are the ones that really define what we're going to do therapeutically, because this is all attempting to think about therapy. So what Jack did was, firstly, he got clinicians to identify features they thought were important, and they would say things like proteinuria or the antibody profile, or maybe having arthritis. And then we also got patients to say the things they thought were important, where they said things like actually the amount of steroid use we've got, or sicker symptoms and things like that. And then there was a review of the literature as well to find things that were important, where you also brought in like the previous molecular analyses, like interferon score, for example, came in. So we got a group of most important variables, and then they were applied in Precise SADS, but instead of just applying a clustering algorithm, so just applying a clustering algorithm can cause problems when you've got too many variables. You can get overfitting so that you get a model that doesn't work on another cohort. So it's quite challenging. So what was used instead was a deep learning based method called a variational autoencoder, which basically you try to compress your data down to a lower number of dimensions in this latent space that's like smaller amount of information, and then you see if from that smaller amount of information can you reproduce what it originally looked like, until you get down to like this bunch of sort of imaginary variables represents a more complex whole, and then validate it in the other data set. So basically, to cut to the conclusion, if you started off with patients who had what we called legacy diagnoses, which were primary Sjögren's, antiphospholipid syndrome, SLE, scleroderma, undifferentiated CTD, lots of those, inflammatory myositis, and mixed connective tissue disease, they all reclassified into something else. So all the diagnoses got broken down and regrouped into something else. And the something else were 5 classes. Some were clinical and some were immunological. So there's one called sicker, which is a lot of Sjögren's, but it also had some other stuff. Some patients came from lupus and UCTD. There's one that's just quiescent disease, which came a bit from all the diagnoses, actually. There's one about active MSK disease, which came again from lots of diagnoses. And then there are 2 molecular ones, one called myeloinflammatory, which is based on a gene expression score, and one called high clinical and high interferon activity. So those last 2 were the most severe, the most sort of multi-organ disease. We did, on the abstract, we thought about breaking that down a little bit more into 7, but actually we've gone back a bit on that and just kept with the 5. But then, so we've basically those 5, they classify more people and they classify with less heterogeneity. So everyone gets a label. You don't have this thing called undifferentiated or mixed anymore. Everyone's in a group. And also when you basically, each of those groups is more homogeneous than the original diagnoses. So lupus is very variable, but the group of active MSK or the group of myeloinflammatory is more homogeneous, both clinically and immunologically. So they should be basket populations for trials. And then Jack also did some long-term follow-up on them and showed that you could predict their long-term outcome depending on the class they were in. Better.
So I really like the looking to the populations that were in trials and the idea that it might change trial outcomes by reclassifying people. It also, maybe I also like it sort of validates a thought I've always had in the clinic. When you're teaching trainees and you have residents in the clinic, we're always talking about it's a connective tissue disease with these features that predict certain things happening. I always try to stop people from calling it lupus or calling it Sjogren's. This is a connective tissue disease with features of ... Because otherwise we get into sometimes a lazy way of thinking where we just treat lupus with lupus drugs, or we just treat Sjogren's and we don't think what's going on. And I think this stuff is fantastic. And as you said, there's other work around this, looking at phenotyping around clusters of autoantibody combinations that Karen Kostenbard has been involved with as well. So there's a whole lot of bit with this, with getting machine learning and deep learning to direct us towards different diagnoses. And I think you're right. You know, it may become time to stop calling things lupus. You know, we're going to call it
because
a different thing.
Yeah, lupus in the skin and lupus in the kidney.
Yeah.
Like, why is that the same disease?
Yeah. and after all, the diagnosis is just based on, you know, intelligent people using clinical acumen to subtype people on the basis of the way it looks, but that doesn't mean it's the mechanism is always the same underlying it.
Yeah, so the next steps are a bit complicated potentially, but one might be that, like, if you did if you took the obvious one would be the active MSK group of people that basically whatever else is going on, their main problem is active arthritis. And you could imagine if you did a trial in that group, wouldn't that be quite an easy trial to do compared to an SLE trial? Because everyone has the same problem. Yeah. The outcome measure can be like a DAS28 type thing, something like that, rather than trying to assess 8 different organs.
Yeah, completely. Well, and it's already supported by the fact that sometimes it's easier to do a lupus nephritis trial than it is to do a lupus trial, because you're absolutely defining the population. You're certain of the underlying pathology, and you know you've got the right patient group.
Yeah, inclusion's easier, standard of care is easier, easier, and the answer will be applicable to more people.
Yeah, so I really like those two. Those were good.
Okay, so the last ones to finish up were some abstracts on nothing to do with new therapies, actually, to do with targets, really.
Yeah, and I think this is the glucocorticoid low disease activity state one, Ed. I think you were looking at that. Yeah,
that's right. So the original definition of LLDAS, which is used often as a target of treatment, is SLEDAI of 4 points or less. None of those points from major organs, none of those points from new activity, PGA okay, and prednisolone 7.5 or less. And that's been quite widely used and very well validated as the more time you spend on that target, the less damage you get, better mortality, better quality of life. But many people have said that isn't the real target of prednisolone 5 milligrams? And therefore, should the LLDAS actually say 5 milligrams? And even the EULAR guidelines kind of say that, actually, that the target's 5, not 7.5. So this is like the original Asia-Pacific sort of collaborators who did the LLDAS looked at this question and basically said, can we tell the difference between people in LLDAS with 7.5 and people in LLDAS with 5 or less in terms of flares, damage, or mortality? And the bottom line is, no, they couldn't tell the difference. They couldn't see any improvement, and therefore the conclusion was you might as well stick with 7.5.
Okay.
So I mean, I do still wonder whether in practice I prefer 5, but perhaps as a validated thing for trials, they couldn't show it.
Okay, so that's not changing. Okay, and then we've got a couple about withdrawal, haven't we? So there are 2 that sort of similar theme. This is the first one. This is from colleagues in Italy. And this was about taking away glucocorticoids in a retrospective study of 500 plus patients. So a number discontinued over time, and they were looking at how many people would flare after the event. I suppose slightly counterintuitively, the people who had their steroids taken away had less flares.
Yeah.
Bear in mind, it's a retrospective study. And in fact, there was a question in the session around that. And I mean, my take home for this is maybe we're really, or clinicians, and certainly the clinicians in Padua, were very good at predicting the patients who were least likely to flare, in whom you could safely take away the steroid. You know, maybe that's what this is showing. They also identified the people that were most likely to flare, and the ones most likely to flare were the ones with low C3. You might predict that. Anti-RMP, would you have predicted that? I'm not sure. Cytopenia.
Yeah.
And vasculitis. So I think 3 of them you would. The anti-RMP, would you have predicted or not? I don't know if I'd have known that for certain. But the other 3 are just disease activity markers.
Yeah, I agree. If you just randomized people to discontinue steroids or continue them, you might have got a different result. But this is what this is showing is the steroid tapering. It was being administered judiciously, wasn't it?
Yeah, absolutely right. Okay, and then the next one. So this is bigger data set. So this starts off with having 4,000 plus patients, and they, as you go through the analysis, it chips away to slightly lower numbers. And this is from the Asia Pacific collaborative cohort of patients. So it's collected from lots of parts of Asia Pacific. And of course, in that population, for anyone that isn't from that part of the world and doesn't know, there are people with very bad lupus in that sort of population. So that's worth just bearing in mind. And they looked at tapering and the individuals that tapered. They looked at steroids being tapered, and they looked at immunosuppressants being tapered. They tried to associate this with the amount of damage, the likelihood of flare, damage accrual over time. And I guess there are a few headline things. They thought that overall reducing steroids didn't seem to be too bad a thing. People didn't do too badly. And in fact, they had some data that they presented around what we know, that steroids, even at fairly low dose in the 5 to 7.5 milligrams range, was associated with more damage in the cohort than those on less. So that seemed to be not such a bad thing. But then they also said that tapering immunosuppressants was more likely to be associated with flares.
Yeah.
And so their message, I think, was you can take away steroids, but when you take away immunosuppressants, be a little bit careful and be thoughtful about which patients you're going to reduce in. And they didn't define in this particular analysis the type of characteristics that would be associated with flaring, but I'm sure they have done or will in the future.
Yeah, again, it's not randomized, so it might just reflect people are making smart clinical decisions. But I kind of thought at the end of it, I think my basic conclusion about how to manage these drugs long term is still the same, which is I like keeping the immunosuppressants, but I like losing the steroids until it.
Yeah, and I suppose, mate, so I agree. And the only thing I say is that I try very hard to get steroids down, but people who've had organ or life-threatening disease, I still leave that 3 milligrams there, even though it seems a tiny amount, until for women, until they're postmenopausal, and I think their disease is likely to become less. Now, maybe I'm being overcautious with my steroid reduction, but I haven't seen anything that tells me I mustn't do that yet.
Yeah, the treat to target trials that are in progress are going to be very important in answering these questions.
Yeah, absolutely.
Okay, so that's the end of our podcast today. So I hope you've all enjoyed this review podcast. Don't forget to visit the Lupus Forum. That's lupus-forum.com. And you can find a great range of lupus content, including slide decks for top research, monthly podcasts, special webinars about key topics of interest. And all these PowerPoints are free for you to download and use in your own teaching and journal clubs. So thank you. And as always, the best of luck in your clinical practice. And thanks, Chris, for joining me.
Thanks very much.